Hello, everyone. My name is Sarah Barget, and I'm the chair for this symposium today. I'm a research geneticist at the U.S. Forest Service Rocky Mountain Research Station in Boise, Idaho, in the United States. And it is a pleasure to have all of you here today. I'm so glad that I could have representatives from all over the world talking about using forbs in restoration today. So the name of the symposium is Restoring Understory Plant Diversity, Considerations when Incorporating Native Forbs in Restoration. And today we have 6 panelists. We have Christina Blandino, who will be talking about regeneration of forbs and seed germination. We have Tara DeCaros, who's talking about seed increase of annual forbs used in restoration in the United States desert, Great Basin Desert. Allison Anury and myself are going to be talking about seed sourcing and considerations when selecting populations for for restoration. And Anita Kermer and Paul Gibson-Roy are going to be talking about their experiences using forbs to restore either ecosystem function or just ecosystem diversity in general. And so I figure we'll go around and begin by doing introductions in that order. So Christina, if you'd like to introduce yourself, that would be wonderful.
Good morning to everyone. My name is Christina Blandino, and I was a PhD student at the Royal Botanic Garden Kew. I was part of an international project, NASEC, Native Seed Science, Technology and Conservation. The objective of, we were 11 PhD students scattered all over Europe, and our aim was to bridge the gap among seed science and small seed producing company to provide the science to increase the production of seeds of wild species for restoration purposes. So my project focused on the forest understory species. So I've studied the regeneration strategies of some species from a temperate European deciduous forest, and trying to define their kind of dormancy and to see if they can group depending on the habitat inside the same forest or depending on other regeneration functional traits. So this is the work I'm presenting here is a review.
You have to open your mic, Sarah.
Thank you, Christina. I really appreciate that. Sorry, I'm trying to unmute myself so that I don't have echo. Next, I would Tara please give a little introduction to her talk and herself?
Sure. So I'm Tara DeCatos, and I work at the University of Nevada, Reno with Beth Ledger. We started working on this project about 14 years ago when I was working for the ARS USDA Agricultural Research Service here in Reno. And I had just moved here, and I was noticing that a lot of the ecosystems around this area the sagebrush has been converted to invasives, but I was noticing that there was this little community of native annuals among the cheatgrass. And so I started to become interested in whether those might be able to be used in restoration, especially in seedings. And so that led to some work that I did with Beth on competition between those native annual forbs and cheatgrass. And then I ended up leaving my position with ARS when I had my 2 kids. And I took a few years off, and then at some point Beth said, hey, do you want to come back and do more work on annuals? So I've been working with her part-time on annuals for the past like 5 or 6 years. And this work has focused more on making seeds available to growers and to restoration practitioners. And so that's what the talk focuses on, is that seed increase work, collecting wild seeds, and then growing them at a very small scale at the university agricultural research station, and then making those seeds available to people who are interested in using them either for experiments or for seed increase and restoration. And I had no background in agriculture or farming, so I probably reinvented the wheel in a few cases, and I learned a lot about how to grow plants in a field. And I enjoyed everybody's talk. I'm looking forward to hearing from everybody. Thank you.
Thank you, Tara. Allison, will you please go next?
Hi, everyone. My name is Allison Anyure, and I also work at the University of Nevada, Reno, in the same lab as Tara with Beth Ledger, and I'm wrapping up my PhD. And the talk that I presented for this symposium focused on sort of identifying different seed sources for forbs, along with a couple other species, and trying to understand where these plants are coming from, how they're shaping them, and how that might impact their restoration down the line. And we're actually, as of yesterday, I went out and collected some Chenactis douglasii, which is a forb that I talked about in here, and we're collecting it for seed increase because it did so well in these trials. So I'm actually really excited to hear about everyone else's experience with forbs, since we're used to working with grasses here in the Great Basin. So I'm just here to learn too. Thank you.
Thanks, Allison. And so I guess I'll go next. I'm Sarah Barget again. I'm a research geneticist with the U.S. Forest Service, but I come from Beth Ledger's lab. I did a lot of my PhD work working with a variety of 10 annual and perennial forbs. And I'm really interested in population level variation and performance of forbs. And so a natural lead into that was working with these forbs in my PhD, and now graduating on to work with them professionally with the Forest Service, conducting a large-scale forb common garden experiment where we've taken seeds from between 65 and 113 populations of 3 different forbs that are of interest because of their value as resources for wildlife. And we've planted them in 6 to 7 different gardens across the West, and looking at performance of them and how that might translate into creating guidance for how we select particular populations for restoration. But what really, there's so many bottlenecks in this process, and I really wanted to talk to people at different stages of the process, because while I'm interested in seed sourcing, I'm also interested in how this plays out on the landscape in a practical restoration context. And so I really like the idea of having this progression of people working at different stages within the cycle. So I hope you enjoyed my talk, and I welcome any questions about the project that we're working on. We're still in the data collection phases, but soon we'll be working on some seed transfer guidance for managers once we have some of our more samples processed. So soon, hopefully. And so next, I'd like to talk to, if Anita could please do her introduction, that would be wonderful.
Yeah, I hope you can hear me. It's so nice to be here, seeing you all, even if it's online. I'm a biologist working at the Anhalt University of Applied Science in Germany, and I'm doing research on biodiversity enhancement in agricultural areas. And at the moment, I'm acting professor for vegetation science, and I'm responsible for several projects dealing with these topics like biodiversity enhancement and use of native forbs in restoration. And my talk was about the idea that since the biodiversity decline is so severe in agricultural areas, we are trying to do some mitigation and using especially native wild plants, native forbs to do something against the pollinator crisis or the crisis of the insect decrease in the landscapes. And we have very good results in installing species-rich vegetation in intensively used vineyards as well as in arable landscapes. We had several projects where we did evaluation of the outcome of using the species-rich seed mixtures and how they benefit, for example, wild bee occurrence on the sites. And we found that it's very important to sow a broad variety of different species, not only to use 5 or 6 or 7. We always recommend to use at least 30 species to have a huge benefit for a broad variety of bees, for example, also the threatened one and the specialist one. And we found a very clear connection to the landscape structure where the restoration sites are. So in simply structured landscapes, we found that the species-rich vegetation we developed benefit mostly generalists, but in more structure-rich landscape, they also benefit rare and endangered wild bee species. Yeah, I'm looking forward to your questions, and yeah, thank you very much.
Thank you, Anita. That was great. And last but not least, Paul, you've been such a great supporter during this whole process, and welcome, and please we'll enjoy an introduction from you now.
Thank you, Sarah. So I'm Paul Gibson-Roy. I'm a manager of ecological restoration with a group called Cowbar Operations, but I'm also an associate with Western Sydney University. I've been involved with grassland and grassy woodland restoration for some time, both as a researcher and as a practitioner. And I'm hoping that my presentation gives people some insight into the constraints and the barriers and the obstacles associated with trying to restore complex, functional, resilient grassland community. So I guess, you know, all those functional components, both the grasses, the forbs, different functional groups, you know, early succession, late succession pioneers, and seeing whether it is feasible, given all those constraints and barriers, whether or not we can put them back together. And so I hope my presentation does give some indication that we have had some success, some small scale success at being able to put those things back together, which is a fantastic outcome. The big challenge for us in Australia is to find the pathways and the incentives to take that knowledge and take that small scale success to the landscape scale that it's needed. So that's sort of what I'm on about. Thank you.
Thank you. Yes, that's always a challenge, upscaling. So we do have one question in the Q&A, and I'll read it out loud, and then we can decide who wants to answer first. How difficult is it to find partners who will grow native forbs? Thinking specifically of the wildflower strips and increasing plots, do you find financial encouragement limiting? So this sounds like maybe a question for Tara and for Anita.
I can say a few words about that. I would say that that's kind of one of the major barriers, is lack of kind of a structure or a system for growers to get into the business, to start growing these native plants, and know that there's going to be a buyer. Because where we are in the Great Basin, most of the seed is purchased by BLM, and the amount purchased really depends on what fires happen and what areas need to be restored, as well as how much budget they have. And so for a grower to convert a field to this type of growing, which is different than other things they might be growing, is really challenging, given that at this point, there's not much of a guarantee that they will be able to make back their money, let alone make a profit. And so that's something that a lot of people have been talking about, trying to find solutions for and ways to guarantee that those seeds can be bought so that growers can build the capacity to start growing those seeds every year.
I can also add something for Europe or for Germany. We have a very elaborated system for seed producing of production of wild seed, and there is a certification system that guarantees that if you buy these expensive seeds, you will really get the wild forms. And since the demand is rather high, because the legislative says you have to use wild seeds when you do restoration or revegetation in the landscape outside settlements. So the demand for the wild seeds is rather high, and so farmers are getting more and more interested in doing this kind of work. Especially in the agricultural land, we have more and more federal states demanding the use of native plants in, for example, the white flower strips agro-environmental scheme. And therefore, the demand for this kind of forbs is rather high. For example, in Saxony-Anhalt, is the federal state where I work, we have in the last 5 years 4,000 hectares of white flower strips established.
Nice. That's great. So we do have another question. This question is for Christina. What kind of strategy would you use for restoring forbs for a temperate forest in a regenerative stand that already has a good canopy, and if a practitioner would be starting from scratch?
Okay, thank you for the question. Well, when the canopy is completely closed, I think that it may be better to not to seed the plants directly, but to grow them in a nursery and to provide plants that already passed the more critical phases of their growth. So plants that are maybe 1 or 2 years old and that have been acclimatized to tolerate shading may be more successful than directly adding seeds in the case of a closed canopy situation.
Great. So I think that was the last question's been proposed. So I have another question that was proposed by one of our panelists and asking me about seed transfer zones and whether increasing. I'm afraid I'm getting an echo. If some people could mute. Christina, could you please mute? Oh. Thank you. Sorry. So the question is, seed transfer zones are increasingly being developed to provide a rational basis for seed movement. Are the metrics used to define seed transfer zones capturing traits that best relate to restoration success? What do people see as the main benefits of developing detailed seed transfer zones, and also maybe unintended negative consequences? So from my perspective, we're looking at performance. We do use plug planted plants in the gardens, and we're looking at fitness related traits, performance related traits in the field, with the idea that variation in those traits will translate into better performance if we can find patterns in performance related to the climates in the places where we've planted them. Perhaps we can predict their performance in different environments and make clear guidance for our managers. But we aren't really looking at specific traits relative to, say, very arid systems or very other challenges that might be experienced at high temperatures or windy areas or erosion or anything. Those aren't the traits that we're really looking at. We're focusing mostly on fitness related traits. We're also incorporating lineage data in there to see if that is a factor. But what I am doing is looking at some early life history traits for these populations as well. So I'm doing some seed germination work and seed trait work as well to look at perhaps how that might be incorporated into these seed transfer zones to look at how establishment might occur from seed. Because we are looking at plug planted plants, we don't know, we wanted to force them to perform so we could get measurements, but I also think it's really important to look at those early life history traits and see whether we can find patterns in dormancy syndromes and things, differences, population level differences in those kinds of early life history traits. I also think it would be great to have other parallel experiments, which we are trying to encourage with academic partners to do work on things related to competition. So we have a partner at a university looking at competition with a major invasive plant across the West. And I also think that looking at below ground traits would be wonderful. Unfortunately, we can't kill our garden plants, and so I can't really ask questions about below ground traits, but I think that those would be really important in the environments where we're going to be using these plants in restoration. So we are going to be trying to look at those questions. I also think that when you create seed transfer zones, these are supposed to be tools for managers, and they're supposed to be easily translatable. So we're trying to create geospatial seed transfer guidance for managers so they can say, oh, if I grow it in this area, these other areas are environmentally similar, and so we should be able to pull seeds from those areas and increase them and use them as successfully. But on the other hand, it can oversimplify things. So there are trade-offs there. It can be really challenging. So I think that once the guidance is created, we really do need to follow up with a test of that seed transfer guidance. We need to go out and do seedings that would mimic what would be happening at a larger scale for a seeding after fire, other disturbance, and see whether we see differences in the performance and the success in those seedings. So I think that's a great question, and it's really, we're just starting out with this, working with Forbes in this way, and I think it's going to be a real adventure. We're going to learn a lot. So we have another question for everyone. What has been your greatest success so far? So, oh, and I see another question that I skipped over accidentally, so I will have to go back to that. But for everyone on the panel, could you please, if you have a great success, could you please share it?
I'll jump in, Sarah, if I may, if I may be so bold. I've been involved in grassland restoration now for probably 2.5 decades, and in Australia in the mid-90s, we were very focused on trying to quantify the loss of grasslands, and we were beginning to understand the degree of threat to them. And our main focus was really on trying to conserve what was left, which is a very understandable perspective. But what we were finding was that even with legislation and restrictions about what people could or couldn't do, we were still losing grasslands, and we still lose them to this very day, grasslands and grassy woodlands. So it became clear that conservation on its own wasn't going to halt their loss. But at that time, the consensus among botanists and ecologists and conservationists was that it wasn't feasible for us humans to restore a grassland that had the same quality, composition, function as a remnant grassland. It was essentially seen as something which was a natural event. I guess for me, my greatest achievement as an individual, and I think as a group of people who have been involved with a group of people who have pursued this for 20 odd years, I think we have demonstrated through really good sort of long-term monitoring of the work that's been done here, that it is actually technically feasible to reconstruct functional, complex, diverse, resilient grasslands. Now, as I said, the challenge for us is to find the pathways and the regulation and the incentives for that to be taken up at large scale, because there is much work to be done. But for me, the great success is to be able to show that that can be done. And now it's up to, our regulators, our leaders, to find a way then to take that knowledge and apply it.
Thank you, Paul. Anita, you're the other person that's really put these things into practice. So would you like to share one of your successes?
Yes. One of our greatest successes is to change the minds of the ministry about using native wild forbs in agro-environmental schemes. And in the last period of the CAP, we were the only federal state who had seed mixtures containing only native forbs, no ornamental plants nor non-native plants. And we convinced also the farmers to use these mixtures because they really work. If they have some do something, not to bring them on the shady sides of woodlands or so, but to use them really on the fields or on the southern exposed sides, or even east or west exposed, then they really work and they bring something positive for the wildlife. We did evaluations not only of wild bees, but also of birds, of farmland birds, and of butterflies. And we have huge effects caused by these mixtures of 30 plants. And we think it's really one of our greatest success that so many hectares were planted with these seeds, and that the farmers really love them because they see the benefits. Other things we are doing is at the moment we try to bring biodiversity back on arable fields. We have a new project starting in August where we will try to do really on a large scale to bring wild plants back in the fields, because the farmers are doing strip till. They have 50 centimeters in between the crop. And we want to bring in these 50 centimeters low growing wild plants, and they developed special machines to so that they don't grow too high and be too competitive. And if this works, this would be a good opportunity to have biodiversity back on a really large scale, because in my region here, the farms are rather big, and the single arable land is about 20-30 hectares. So maybe same in the United States, but not in other European countries.
Nice. Thank you for sharing, Anita.
Yeah,
so I would say, my project is still in the works. We're still collecting data, but I would say my biggest success right now is just realizing how much support there is out there for using forbs in restoration, and all of the partnerships that I've been able to work on with different people as different aspects of this project, with the Seeds of Success group to collect the seeds, and BLM land managers and Forest Service land managers that are willing to let us put gardens in on public lands, and the researchers from universities that we're working with, in the nonprofits. It's just been this huge collaborative effort, and all of that support has just really inspired me to know that people want this information, and it's been wonderful to have that feedback. Would anyone else like to share a success? Smaller, big or small?
I'll just share a small one. I was kind of amazed when we started growing the seeds. We never used any irrigation, and I was just amazed that we were actually able to grow almost all of the species almost every year successfully with no irrigation at all, which to me was kind of very suggestive that they could be effective in restoration, because low precipitation is our main challenge in doing seedings. So.
And Tara, I would add that all of your experimentation with seed collection, like getting the seeds off of the plants, you had a lot of ingenuity. I know it's tricky.
There were some bad ideas tried at first, and they improved over time.
So I'm going to move on to the question that I accidentally skipped over. It says, I have been struggling with establishing a mix of native grasses and forbs in urban natural areas and parks. I struggle with the forbs since typical establishment operations require mowing to keep the weeds down, while the native grasses establish over 3 years. By the time the grasses have established, the forbs are not present typically. What sequence would you recommend for establishing a mix? I assume a mix of seed, forbs and grasses.
Yes, I thought Paul would like to answer the question. I was just typing an answer to this question, but it seems to have vanished at the moment. So I wanted to say that it depends on the climate zone you're working in, and I just can answer for Central Europe conditions. So I need to feedback where, yeah, for what climate zone the question was asked.
I'm going to get back to that, Caitlin. If you could let us know what system, your climate system you're working in, that would be wonderful, and we'll come back to that. I'm going to go to another question for Paul. I was just interested in Paul's approach to strip some topsoil to bring soil parameters back into the range of native species. I find that soil is underappreciated. What are the approaches of other panelists to match plants to soils? Okay, so it's for everyone, but it's about, it was spurred by Paul's research using, with his techniques with soil. So have others thought about soils in their experiments and research? I can say that we are partnering with a soil scientist at the Utah State University, and Andrew Komatiski, and he is working with a graduate student of his to take soils from all of our different gardens and look at how microbial communities may affect performance of those plants. So we are doing some aspect of looking at soils and their influence on performance, but we're not actually incorporating the analysis of the soil into the seed transfer guidance. And it's just a little bit challenging because the scale of variability in soils is so fine. It can be really difficult to deal with that on the scales that restoration is happening. But I don't think it's impossible. I really do think soils are very important, and I'd like to partner with more people who know more about soils than I do. I know I'm a plant person, and the obvious thing would be the way we'd be thinking about soils, and we do, but not to the degree that people that are experts in soil think about them. So I do want to hear more about how others are incorporating that.
Look, perhaps I might make just a comment, Sarah, just while other people then are thinking about that topsoil stripping. It seems like quite a radical approach, and I know some years ago I was over in the States and was visiting restoration in the Midwest, and most of those people thought I was crazy when I was talking about removing topsoil. But in Australia, really, our arable landscapes, the soil characteristics are radically different under agricultural conditions to what they were under remnant or pre-agricultural conditions. So it didn't really dawn on us for a long time, and what really sort of alerted me to even the prospect of doing this was often I would be driving through our landscapes and look at historic roadsides where we were seeing these lovely little remnants on these roadsides, but the remnants themselves were artifacts of earlier road clearing where they'd stripped off soils to lay down material while they were building the roads, and taking away that topsoil meant that they had taken away, as I said in my presentation, a lot of the nutrients which have been applied in those landscapes for agriculture, but it physically removed the weed banks. So when we started testing that, we found that that was a radical shift in our ability to put back our subdominant natives onto a soil that then all the exotics, which eventually will come back in there, meant that they weren't able to dominate. They were then just subdominant components. And that was a a huge shift forward. Now, obviously in Australia, we're not going to be arguing that you're removing topsoil over millions of hectares of landscape to restore grasslands. But in Australia, the size of our best grassland remnants is often no larger than a few hectares. So it's feasible for us to restore them at that scale by removing topsoil, because that's the scale of the remnants that we have left. And many people were very nervous about what scalping would do, say, to soil microbial communities. But we've done research down the track on our restored grasslands, and we've found that the degree of mycorrhizal relationships, for example, are no different in our scalped and then restored grasslands to what they are in remnants. So it seems that what seems to be a dramatic upfront soil preparation technique in the longer run enables us to establish complex vegetation that then is able to reconnect in terms of the below ground biota. But also, as Anita was talking about in her European restorations, what we find is that these complex grasslands then are absolute magnets for invertebrates and reptiles and other sort of things. So they become very complex at a multi-trophic level. So I think understanding the soil is a very important component. And I know that people in Europe, for example, have also, tinkered with topsoil removal as well to try and reduce nutrient loads. But I understand that in America it isn't as prominent, except once when I was in California talking to restorationists, they did seem very interested in at least that concept. So it's obviously very specific to where you are in the world. But soils are a very important component of putting plants back successfully.
Thank you, Paul. We do have for Anita, we do have Caitlin offered up that she's working in Calgary in an AB semi-arid climate. Are you familiar with that?
No, unfortunately not. But I could answer the question from an anonymous attendee about, if you like.
That would be great.
Yeah, we did some research in this topic to sow only parts of a site and wait if the species migrate to the other non-restored site. And this is rather difficult because if there is no establishment or germination niche on the site, you will get no result. Our experiments are now 11 years old, and we had this extremely dry summer in 2018 and also a dry one in 2019. And this caused some gaps in the sward. And in these gaps, the species, the forb species, the target species, established in these grass swards. But if you don't have disturbance events, it also could be some fox or some other wild animals digging on the side. If the diaspore pressure is high enough, and if you have some disturbances on the side, then this might work. Fortunately, we don't have so many invasive species on the grassland. So there is in Germany, there is at the moment no real problem with open grass swards and lots of invasives coming in as it is in the United States. But this process of spontaneous succession is really, really slow. And if you want to have a fast result, then you always have to do destruction of the grass swards and to bring the seeds of the target species in to the site.
Great. Does anybody else want to tackle that question. It was, I know very little about grassland restoration, so let's bring it back to the basics. When you are able to restore a few acres of native grasses, are the plants able to spread into adjacent areas, or do you find that due to the pressure of invasive species and weeds, that it is hard to maintain the size of grassland? So thank you for your answer, Anita. That's great. And I didn't know if anyone had anything else to add.
In that situation, Sarah, I think if you've established a plot of grassland, you can manage their expansion by, for example, reducing the dominance of the vegetation to create gaps around that. So that can be either by spraying out the vegetation that you want it to expand into, by, as Anita said, cultivating and creating the gaps to do that. But it does often take active management to entice them out beyond where you've established them.
I did have another question for you, Paul. People asked, what do you do with the soil that you take off from the top in your treatments?
It's quite simple. You're aiming not to move it very far, because if you're putting it in a truck and taking it somewhere, that's very expensive. So when I'm working on a farm, all I physically do is take that topsoil from where I've stripped it and just move it across to the area adjoining it. So essentially I'm taking the nutrients to just across to where the farmer can still use that topsoil, and so they can still be producing pasture or crops there, but I'm taking it away from where I want it to restore the native. So ideally you're not moving it very far, or you can invert it, so you can flip it upside down and bury it. So there are a lot of different ways to do it, but ideally you take it to where you want it, or you don't take it very far, because that's expensive.
That makes a lot of sense, yeah. We have another question. How concerned are you about non-specific selection based on traits that allow plants to grow in cultivation? For example, loss of genetic diversity. Maybe Allison, do you have an answer along those lines?
Yeah, so something that we've done is not focus like a plant breeder, where you take you find a plant that you like and then expand and cultivate that single plant with the traits that you like, but instead work on population level traits. So then sort of what is the average trait values over an entire population, which could be 500 plus plants. Generally, that's the size that we work with, at least. So then we collect always from these huge populations to try and maintain that genetic diversity. But then something we're also doing, for instance, with these plants that we're putting in increase, where we collect the seeds, and then from a portion of those seeds, we're going to hold off on some of them and then grow those out and then check their genetic diversity in sort of different traits to see how going through the cultivation process is changing over time. So then something we're going to be doing is collecting from each level of cultivation and over a couple years, and then compare them, because that's really the only way to know if you're actually losing any sort of trait values or diversity over time. But that's something we're doing over the next 5 years. So yeah.
Thank you, Allison. Thank you for that answer. It looks like we have a few more questions in the Q&A, but I do believe that we are out of time right now. So I just want to make sure people have time to get to their next session. I do want to take a moment to thank the panel. Thank you so much. You've been very supportive, and it's been a pleasure working with you all. And hopefully I get to meet you in person at another SCR conference in the future or at other opportunities. And thank you to everyone who has participated in this Q&A session. It's been wonderful to hear these questions and have a chance to talk amongst ourselves about the challenges that we're facing and the successes too. So thank you so much. And I'm not sure if there's a formal way of wrapping up the session, but I appreciate it, everybody. All right, thank you.